Step 1, EMP. Step 2, Krebs cycle. Step 3, electron transport chain. If we are asking what is the differences, what is the similarities of aerobic respiration and anaerobic respiration, you say all the things in EMP, in Krebs cycle, in ETC, in both of them are the same. If asking what is the differences between aerobic respiration and anaerobic respiration, you say at the end of ETC, if oxygen is electron acceptor, it is aerobic respiration. Is a matter other than oxygen, like phosphate, sulfate, or the other things are accepting electron, it is anaerobic respiration. It is anaerobic respiration. For all of things, many enzymes are necessary. Many enzymes in EMP, many enzymes in Krebs cycle, many enzymes in ETC. All organisms have EMP because they have enzymes of EMP. Aerobic respirators has the enzymes of EMP. Anaerobic respirators has enzymes of EMP. And fermenters, those organisms which can only do fermentation, they have the enzymes of EMP. But in fermenters, there is no enzymes of Krebs cycle. There is no enzymes of electron transport chain. So in fermentation, we have only EMP. If we asking you what is the similarity of fermentation and respiration, you say only EMP. Only EMP is the common part of fermentation with respiration, because in fermentation there is no Krebs cycle, there is no electron transport chain. Okay, so you think anaerobic respiration is more similar to aerobic respiration or fermentation? Who can help me? Anaerobic respiration is more similar to aerobic respiration or more similar to fermentation? Who can answer me? Yes, I do it. Very good. When you see during the explanations, many times I explain one thing, so it is showing you the place of questions for exam. Many times I repeated some questions, so those things which I want to give you in exam, many times I repeated, so you understand which parts are very important. Yes, aerobic respiration and anaerobic respiration, both of them has EMP, both of them has Krebs cycle, both of them has electron transport chain, but in fermentation, there is no Krebs cycle, there is no electron transport, so anaerobic respiration is more similar to aerobic respiration than to fermentation. What is the similarity of anaerobic respiration and fermentation? Who can tell me? What is the similarity of anaerobic respiration and fermentation? Okay, and the other thing? The other thing? Yes, very good. But the other thing? The other thing is that both of them are working in the absence of oxygen. Both of them are working with the absence of oxygen, yes? Fermentation and anaerobic respiration. So do you think anaerobic respiration produce more ATP or fermentation produce more ATP? Which one produce more ATP? Anaerobic respiration or fermentation? Very good. Anaerobic respiration. Why? Because many ATP in AMP, many ATP in Krebs cycle and electron transport chain. But here in fermentation, very low production of ATP we have. Very low production of ATP we have here. Which of them you have in your body, in your cells? Do you have aerobic respiration, anaerobic respiration, or fermentation, or all of them, or some of them? Which one do you have in your cells? Aerobic respiration, anaerobic respiration, fermentation? Microbes, bacteria, all the living things needs energy. For energy production, we told you we have three ways. Energy production by aerobic respiration, energy production by anaerobic respiration, energy production by fermentation. You are a living organism. Your cells needs energy. You solve sugar in your tea. You eat starch, and your body use glucose, starch, and the other things. They put it on which way? Put it in aerobic respiration, put it in anaerobic respiration, or put it in fermentation? Aerobic respiration. Very good. Mrs. Zuhair. Very good. Aerobic respiration. So what is the use of many times breathing? Even though when you are in sleeping, you are breathing. You have inhalation. You need oxygen. What's the oxygen used in your body? First EMP, you use breaking glucose, sending acetyl coenzyme A to Krebs cycle, and sending NADH2 and FADH2 to electron transport chain, and producing ATP. ATP for what? For working heart, for working brain, for working kidney, for walking, for running, for speaking. What do you need? You need ATP. Okay, Mr. Sajjad Ittad Abbas. The question is that we have three ways for energy production. First, aerobic respiration. Second, anaerobic respiration. Third, fermentation. In all organisms, we as a human, which of them we are using? Aerobic respiration or anaerobic respiration or fermentation? Yes, we are aerobic. We need oxygen. We need oxygen for what? We need oxygen for the end of electron transport chain. We need it to give it with breath and sending oxygen to all of our cells in the mitochondria at the end of electron transport chain as an electron acceptor. If 5 minutes not breathing, what's happening? You will be on an anoxic phase. Anoxic phase. What's its meaning? If your cells of brain, heart cells, lung cells, kidney cells, and the others won't receive sufficient oxygen, so electron transport chain stopped. So ATP production stop. With no ATP, brain not working, heart not working, kidney not working, what's happening? Die. The person die. Of course, no. Don't breathe 5 minutes. We cannot continue the life with anaerobic respiration. We don't have anaerobic respiration as a human. Now the question is that, do we have fermentation? Who can answer? Do we have fermentation in our body? Anybody knows? Do we have fermentation in our cells? We humans, we are human. Do we have fermentation in our cells? Okay. No, 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 and yes. Mr. Sajjadi thought yes is okay. When do we have fermentation? Suppose when you go to gym or you go to a sport, you have a guide. One person tell you how to have exercise. He tell you have aerobic exercise. He show you how to do aerobic exercise. What does it mean? He say you as much as have exercise, which oxygen, sufficient oxygen your cells can receive. If you are running very hard, if you are running very hard for 1 hour, what happens to your foot? Why you have foot pain? Why your muscle? Why do you have muscle pain? Why do you have muscle pain? Because when you order the cells running and running and running, and oxygen is not sufficient, what they can do? They continue the work without oxygen and going to the fermentation and little ATP production. So lactic acid production happening in your foot. So you have foot pain. And you should have a rest for a few hours or a few days, like a football play. After football play, many days or some hours, you should have a rest. These lactic acids coming from fermentation removing. But if you have aerobic respiration, if you have exercise, as much as oxygen is present in your tissues, after exercise you are fresh. But if you do exercise very hard and the oxygen necessary the cells are limited, after exercise you are tired. Okay, so normally we don't have and we don't like to have fermentation. But if you use your body more than it is necessary, your body goes through fermentation and you will be tired. Okay, you get the point? All of you? Okay, good. But we are not fermentator. We are not fermentator. We are oxidative organism. We are aerobic. But if we force ourselves, they don't have another way. They do it by fermentation. Okay. We explained you the EMP. We explained you the other ways similar to EMP. We explained you the Krebs cycle and ATP and the other things produced. Here, again, we can see it is mitochondria, like in our body. Why do we need oxygen? Because the final electron acceptor is oxygen. If there is no oxygen, no ATP here, no ATP here, and no ATP here, and without ATP, we will die. We will die. Okay, it is mitochondria. Yes, aerobic respiration. It is what is present in our cells. Aerobic respirations in human. Some bacteria completely look like human cells, like Azotobacter vinelandi, obligate aerobes with oxygen. They continue the life. Without oxygen, they are killed. They have aerobic respiration, but more complicated. In human, we have only one cytochrome oxidase. In human, we have only one cytochrome oxidase. But in bacteria, we have different part, different terminal of cytochrome oxidase. You can see cytochrome oxidase O, cytochrome oxidase D, cytochrome oxidase A1. Different cytochromes are present. But this is aerobic respiration. And without oxygen, cannot continue their life. What's about this one? This is facultative anaerobes. Facultative anaerobes. What does it mean? It means if oxygen is present, you say welcome and do it with aerobic respiration. If oxygen is not present, say welcome to nitrate, or the other things, and going to anaerobic respiration. Going to the anaerobic respiration. So this bacteria, in the presence and in the absence of oxygen, can live. In the presence of oxygen or absence of the oxygen, this bacteria can live. And we say this is facultative anaerobe. It is a facultative anaerobe. With oxygen and without oxygen, no difference. It is very well continue their life. But this time, like us, like human, with oxygen, have life. Without oxygen, will die. Okay? As you see now, till this lecture, it is repeating till now, yes? Not a new things. So whatever I'm repeating, it is showing you where is the questions of the exam. What's repeating many times, so it is important. So have some marks on them. So anaerobic respiration, As Mrs. Isra Akram, as I told you previously, if we explain a car without a picture, how can we understand? If we show you a car picture without text, how can you understand? So the text and the figures are with each other understandable. With each other, they can be understandable. Whatever you cannot understand, ask me many times. If 1,000th time, ask me 1,000th time, I explain you. And you have a great thing in your home, it is internet. Sometimes you should go through it and try to understand. This is the difference between high school and the university. In high school, when the lecture finished, all the things finished. In high school, when the lecture finished, all the things are started. Okay? Anaerobic respiration. In anaerobic respiration, final electron acceptor in electron transport chain is nitrate, nitrite, N2O, sulfate, carbonate, and etc. So I showed you these things in the photo. When you see the photo, you understand these things. When you read these things, you understand the photo. Both of them are in the parallel way. Occurs in strict anaerobes and facultative anaerobes. So these things in strict anaerobes and facultative anaerobes can be seen. Sometimes Pseudomonas species, different species of Pseudomonas, and some species of Bacillus can use these things like nitrate as the final electron acceptor. ATP generation varies from bacteria to bacteria and always less than aerobic respiration. So, first choice, if possible, is aerobic respiration. Second choice, if possible, is anaerobic respiration. And the last choice is fermentation, Alternative electron carriers are used in ETEC. So in anaerobic respiration, those microbes which cannot use oxygen, or facultative anaerobes which oxygen is not present, they can use nitrate as the final electron acceptor. By positive charge hydrogen, it will change to nitrite, changing to nitrite. You remember in aerobic respiration what happened? In anaerobic respiration, we have oxygen here instead of nitrate, and here we have CO2 and H2O. CO2. But when the final electron acceptor is nitrate, instead of CO2, we have nitrate. Or instead of nitrate, some bacteria use sulfate instead of oxygen as an electron acceptor. With positive charge hydrogen, we have this bacteria produce S2 negative charge plus water. But this nitrate is toxic also. So they must use nitrate as electron acceptor, but this thing is not good. So what's happening to that? They should change, they produce nitrate, and this is toxic. We want to see what happens to this. Many bacteria have electron transport chain. It says many, not saying all. So those one have electron transport chain, they have respiration. Aerobic or anaerobic. But what's about those doesn't have electron transport chain? I told you they have fermentation. So if electron transport chain present, respiration. If not present, fermentation. Can operate with exogenous electron acceptors other than oxygen. Aerobic respiration, electron acceptor oxygen. Anaerobic respiration, electron acceptor other than oxygen. It is not new things. All of this I explained. Now we are reading only. This energy yielding process is called anaerobic respiration. The major electron acceptors are nitrate, sulfate, CO2, but metals and a few organic molecules can also be reduced. Some bacteria can use nitrate as the electron acceptor at the end of electron transport chain. So some bacteria can use nitrate in ETC and still produce ATP. And producing ATP, it's okay. Often this process is dissimilatory nitrate reduction. This is dissimilatory nitrate reduction. Nitrate may be reduced to nitrate by nitrate reductase. It is an enzyme. Nitrate reductase, which replaces cytochrome. Okay? So I told you nitrate is toxic. So what's happening to that? This bacteria, after using this as electron acceptor, changing nitrate to NO and changing NO to N2O and changing to an inert gas, not toxic. So bacteria using for anaerobic respiration nitrate using produce toxic substance, but this toxic substance can be changed by them to an inert gas and non-toxic gas. This interactions we say is denitrification. Denitrification. If asking you, what is the denitrification? You say, when nitrate using as electron acceptor and nitrate is produced by some enzymes, this toxic substance will change to nitrogen, non-toxic substance, nitrogen. Changing nitrate to nitrogen, we say is denitrification. Each nitrate will then accept electrons and the product will be non-toxic. The product is it is summarized and it is in detail. It is summarized and it is in detail. Denitrification is a multi-step process. It is a multi-step process with 4 enzymes. 1, 2, 3, 4 enzymes for these 4 interactions. Participating nitrate reductase, again nitrate reductase, nitric oxide reductase, and nitrous oxide reductase. Many reductase enzymes are used for changing nitrate to the final product as non-toxic nitrogen. This is denitrification. This is denitrification. Is it simple? You get it? Okay? Very good. Thank you, Mr. Hussein, Muhammad Hassoun, Mrs. Zahra Zuhair, Mr. Sajjad Ittihad, and Mrs. Akram and Mr. Hassan Khaled and Zaram and Sajid and the other friends. So now we come to the other side. If an organism doesn't have the enzymes for Krebs cycle, doesn't have the enzymes for electron transport chain, so how continue the life? By fermentation. By fermentation. It is not an economic way, but it's okay. For life is okay. Fermentation is the least energy production. Respiration is maximum production, and it is the least production. Who can remember how many ATP come from one molecule glucose in the aerobic respiration? Who can remember? Last time we calculated. How many ATP comes from aerobic respiration? 38. Very good. Mr. Sajjadita, thank you. It shows you study the lectures after each lecture, and Mrs. Shima Uda and Mr. Hassan Khaled, Zarqam, and all of you are good. Excellent. One part of work is the work class, so I give maximum score for class work because you are good students. Okay, by the way, theory 8 we had from respiration. I want to show one thing here. See, what is this? This is EMP. This is EMP. The first part for respiration. In fermentation, these two parts are the same. But from EMP in respiration, it goes to the second round. But in fermentation, all the things finished over here. All the things finished from here. What does it mean? Who can remember how many ATP we have from EMP? How many ATP? I show you the position here. One is here, one is here, and one is here. This is production, and 2 reduction. How many ATP is produced in the EMP? Very good. Excellent. Excellent. 8. I show you. We have 2 molecules here and 2 molecules here. It is 4. And 2 molecules NADH2. 2 molecules NADH2. It is 6 ATP. Yes? 2 here and 2 here. 6 plus 4. 6 plus 4 equals to 10. 10. But 1, 2. 2 removing. 2 consuming. So 10 minus 2 equals to 8. We have 8 in EMP. And 30, you remember, you told me 38 in total. And 30 from Krebs cycle and from the bridge between EMP and Krebs cycle. Okay? But what's about here? In fermentation, see glucose changing to pyruvate in fermentation. till here are the same. Glucose, glucose 6-phosphate, fructose 6-phosphate, fructose 1 and 6B phosphate, glyceraldehyde 3-phosphate, 1 and 3 D-phosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and pyruvate. Till here are the same. Only this different. Here pyruvate change to acetyl-CoA and go to Krebs cycle, but here pyruvate change to an organic molecule like lactic acid. Like lactic acid. And all the things finished in fermentation over here. But in respiration, all the things start here. Going to the Krebs cycle, second round, and going to the electron transport chain as the third round. Okay? Now we see here we have an NADH2 production and 2 ATP here and 2 ATP here. 2 ATP from glucose to pyruvate and 2 NADH2 from glucose to pyruvate. But when pyruvate changed to acetaldehyde and ethanol, again NADH2 is removing, using, consuming. So whatever produced, consumed. So only in fermentation we have 2 ATP. How many ATP in fermentation? How many ATP in fermentation? Only 2. Which one is economical? Respiration or fermentation? Respiration is better or fermentation is better? Very good. Of course, respiration is economical. Fermentation is not economic. So what can we do? For example, you think 2 friends have 2 cars. One car they go the same way by 2 cars. One car used 20 liter petroleum, the other used 40 liter petroleum. Both of them are car, but one is economic and one is not economically. But what can we do? That person doesn't have enough money to change it. Some bacteria have this phenotype, have this genotype, cannot do anything. This is what they have. They should use it. Okay, they have, with 1 glucose in fermentation, they receive 2 ATP and continue the life. This is okay. They cannot do anything. They should continue the life. But in respiration, from 1 glucose, 38 ATP coming up. So it is very economic. Then fermentation. Then fermentation. Okay, now we read what we told you. Now we read what we understand before reading this. In the absence of aerobic or anaerobic respiration, NADH is not oxidized by the electron transport chain. This is because no external electron acceptor is available. But in continue to the metabolism, NAD must be regenerated. In such a situation, microorganisms don't convert pyruvate to acetyl-CoA. Pyruvate won't change to acetyl-CoA. It changed to an organic molecule. It changed to organic molecule. Instead, they use pyruvate or its derivates as the electron acceptor for the reoxidation of NADH. What NADH produced, again consumed by reoxidation. It also leads to production of ATP. So only from fermentation we have ATP. NADH, whatever produced, consuming. So forget it. There is no NADH. This process of extraction of energy from organic substrates in the absence of oxygen is called fermentation. So if asking you what is the process of fermentation, you say this process of extraction of energy from organic substances in the absence of oxygen is called fermentation. The process of fermentation include conversion of NADH to NAD, whatever produced consumed, so it is no use in ATP production. Generation of ATP, it's okay to ATP. It is only for to have life. An electron acceptor, which is usually pyruvate or its derivates, the organic matter produced is the electron acceptor 2. Is the electron acceptor 2. In biochemistry, fermentation is an enzyme catalyzed energy generating process in which organic compounds act as both donors and acceptors of electrons. The lactic acid, the ethanol, what a organic molecule produced is the electron donor and acceptor. In microbiology, fermentation, which involves the conversion of substrates to desired product with the help of microorganisms. Okay, so in microbiology, we use fermentation because, for example, we want to have alcohol. In hospitals for disinfection, before injection, in these days, in Corona days, in COVID-19 days, you have alcohol to disinfect your hands, to disinfect your instruments. So from where you receive alcohol? Alcohol producing by microbes by fermentation. So now you understand why we should know the metabolism. If we want to use a microbe and we want to give order, for example, you go to a restaurant and give an order to the people there. They go to the kitchen and bring you whatever you want. If you want a bacteria or a yeast do some job for you, first you should know that. After knowing that, give an order to that microbe to do it for you. Who knows what is the science of using of beneficial microbes? What is the science of using the beneficial microbes? Anybody knows? Okay, repeating. For example, who knows diabetics from which source received insulin? Have you ever seen a diabetic person? His life is dependent on insulin. Who prepare insulin for this person? From which source we receive insulin? Okay, I'm waiting for your answers. Many students are writing. Please, short answer. Alcohol fermentation is anaerobic. Yes, of course it is. I don't understand the question. Okay, you want to go to a drugstore to get insulin if you are diabetic. Drugstore from which source receive insulin? Who produce insulin? How can you make insulin? For example, you want in Iraq, you have an industry for insulin production. What is the way? Did you understand the question? Again, repeat the question. See, if I want to produce, to sell the milk, what can I do? I should have some places, I should have some cows, some sheep, and the other things, and get milk and sell it. We have some persons need milk. Now some persons need insulin. Do you know insulin? This is for glucose in the blood. Now I want to have a place to make insulin. What is the source of insulin? How can I bring insulin for these people? How can make and sell the insulin? It is not related to lactic acid. It is not related to, from animal, it is very difficult. It is very difficult. Purification is very difficult. It is very costly, high price, and sometimes are infected with viruses and the other things. And this insulin sometimes with origin of animal, sometimes make the patient neurological problems from animals, not possible, but very problematic, very side effects. Okay, very good. Mrs. Zahra Zuhair, excellent. See, many items today are produced from bacterial origin. From animal, not possible. But E. coli doesn't have the phenotype of insulin. Insulin is a gene located in the cell of human. E. coli doesn't have it. How can we send insulin gene from human to the E. coli? What is the tool? Our tool is genetic engineering. Genetic engineering. This is our tool. By genetic engineering, we can transfer the insulin gene from a human to a bacteria, and in industry, this bacteria produce sufficient insulin. You get it? We can transfer a gene from a human, from pancreas, from a human to a bacteria, then give substrate to the bacteria in industry, and in industry, this bacterium produce insulin. Okay, you get it? Okay, repeat which part, many items I told you. See, sometimes we want a bacterium do one thing, it is not in its genotype. Sometimes we want one bacteria do one thing which doesn't have the necessary gene. Sometimes you want a bacterium like E. coli produce insulin without having the gene. So it is easy. We can get the gene of insulin from human and transfer it to a bacteria like E. coli, and E. coli produce insulin. You get the point? What is our tool? It is genetic engineering. Do you get? Okay. Today, in all drug stores you go and buy insulin, it is by bacterial origin, not animal origin, not human origin. The insulin is present in your drug stores in UK, in USA, in Japan, in all the world. So you see, bacteria are so kind, very helpful. Okay, so what is this? This is biotechnology. What is this? The use of organisms for beneficial purpose for human. The use of organisms, the use of animals, the use of plants, the use of microbes, the use of living things for beneficial purpose for humans. This is biotechnology. If in biotechnology you use microbes, it is microbial biotechnology. If in biotechnology you use microbes for lactic acid production, for methane production, for butyric acid production. Do you have any another biotechnology? Do you have Murtaza Sajil, correct? E. coli today is using, for example, in Iran or in Iraq, we want to produce insulin. So we should transfer the gene of insulin to E. coli, and then sufficient quantity. How can we reach to sufficient quantity? By industrial microbiology. So if in lab, very small insulin I'm producing, so what is the use of it? In lab production, it is not sufficient. When you want to have a production for a country, for the world, you should go through the industrial microbiology. So when you have an industry for insulin, it is industrial microbiology. You have industry for alcohol production. You have industry for antibiotic production. You have industry for food production. Here is student Wassam, Jasem. Bacteria make insulin. Yes, of course. E. coli produce insulin for us today. If we didn't use E. coli, many people died by diabetic disease. We have industry for cheese. How can we make cheese without help of microbes? How can we make yogurt? Are you hearing me? Are you hearing me? Okay, I suppose disconnected. How can we make cheese? How can we make yogurt? How can we make many foods without microbes? So now your view to the microbes are different than previous. Previously, you know all the things are pathogenic microbes, but very little of microbes are pathogenic. Most of them are useful for us. Most microbes are useful and we use them. For example, how can we prepare vinegar? You know, vinegar without help of microbes. And many other things, antibiotics, vinegar, and alcohol, lactic acid, antibiotics, many things are producing by microbes. So how can we produce them without knowing the metabolism? Now, all of these help us to understand why should we know the metabolism. How can ask a bacteria to produce antibiotic without knowing its metabolism? How can we ask a bacterium to produce vinegar without knowing the metabolism? So now you understand. These things is not only classical items only for to have a score and finishing the class. You will be in two groups. One group only get the degree and the other group using the degree. Using the degree when happening to understand the concepts. To understand the concepts. Okay? So by fermentation, different fermentation happening and different things can be produced. I see till okay, one page. This page we discuss and we finish it. Alcohol fermentation. Most of the alcohol fermentation in the nature and in the industry is carried out by yeast belonging to the Saccharomyces species. These organisms ferment sugars to ethanol and CO2 by the following equation. C6H12O6, a sugar like glucose, like sucrose, or the other things, and changing it by fermentation to organic matters like ethanol or lactate or the other things plus CO2. Yeast employ EMP. I told you EMP is the common part in all the energy production. Yeast employ EMP pathway for glucose breakdown. The pyruvate produced is decarboxylated to acetaldehyde, which is then reduced to ethanol. by alcohol dehydrogenase enzyme. The NADH produced during the glycolysis acts as the electron donor. Only 2 ATP are produced per molecule of glucose during this fermentation. All the things are repeating. Yes, many times I discussed it before in the text. Some strictly anaerobic bacterial species also carry out alcohol fermentation, like Zymomonas mobilis, Sarcina ventricoli, Erwinia amylovora. However, they also form small quantities of other products like acetate and molecular hydrogen, lactate, and etc. I explained you all the things of fermentation and EMP are the same. Only difference is here. After pyruvate, we have an organic molecule, and this NADH2 produced consumed here. So no NADH production, and incomplete only we have 2 ATP. Some bacteria and some yeast produce only lactate. So we say they are homofermenters. Homofermenters. Only 1 minute, we will finish. Hussein, Sabah, Saddam, I am tired too. So in 1 minute, all of us, we go to have a tea with biscuits, okay? But some microbes, lactate and the other things, we say they are heterolactic. Heterolactate fermenters. But this is homofermenters. I explained this, only ATP production in fermentation, because whatever NADH2 produced, consumed, and we have only ATP. Okay, next week on Tuesday, like previous Tuesday, we continue the lecture. Don't forget. No, next Tuesday, no, tomorrow. Tomorrow, Friday, like Tuesdays, we have a lecture. Don't forget it. Tomorrow, Friday, the time like today, we have lecture, and we, with help of each other, we finish this semester successfully. Okay, 50% here is ready. What's about your 50%? You should try it. If any question, you can ask me.